Good morning, good afternoon, good evening. Welcome to your day 2 of the RxODE Workshop, a brief hands-on introduction to PMX Simulation with RxODE. We are really excited to share that we have had an overwhelming response and have close to 500 participants from all over the world interested in taking this workshop, ranging from Brazil, Chile, India, Japan, Malaysia, Singapore, South Africa, Spain, United States, and many, many other parts of the world. Thank you for being here, and thank you for giving us the opportunity to share this knowledge. Today's workshop is hosted by Project Dontabhaktuni and ISAP and is sponsored by Pharma Pro Consulting and CRC Pharma. I'm your host, Aruna Dontabhaktuni. Something about me. I am the founder and CEO of Pharma Pro Consulting and the founder and scientific director of Project Dontabhaktuni. Prior to starting my own company, I was a director at Lilly, and I headed the clinical PK/PD and pharmacometrics group there. In my career, I have supported 60+ compounds across multiple disease areas, from preclinical to registration and approval. So a couple logistics are that all the participants in this workshop will be muted. However, you can reach out to us, and if you have any questions, you can write to us using the question and answer button, either at the top of your screen or at the bottom of your screen. And we will answer your questions in the order we receive. And if we are not able to address the questions during the workshop, we will do our best to get back to you after the workshop. So something about Project Dontabhaktuni. This is a non-profit organization created to support scientists, students, postdocs from around the world, supporting them in growing their career by providing them access to workshops, training programs, and mentoring programs. Now, how can you support us? You can support us by joining hands with us, helping us in participating in these workshops, take advantage of the mentoring programs, take on being mentors in these programs, and of course, by spreading the word. And so is our motto, empowering you to empower others. Last year, again, Project Dontabhaktuni has been on only for a year, and last year, a few of us had visited India and China, had multiple workshops, reached out to 300+ scientists during these visits. And this year, as the world is dealing with the COVID-19 and the lockdowns, we have chosen to do our workshops online. So our next workshop is also going to be a 2-day hands-on workshop. The topic is how clinical pharmacology shapes the strategy of clinical drug development from discovery to registration, which is going to be conducted by Partha Nandi, Senior Director, Clinical Pharmacology and Pharmacometrics from Janssen. And the dates are August 19th and August 26th. Now, without further ado, let's get to our today's workshop. I'd like to invite Dr. Wenping Wang, who is a distinguished scientist. He has worked in both consulting companies as well as pharmaceutical companies in his career, ranging from Farsight, Johnson & Johnson, and now at Novartis. He is an executive director of pharmacometrics at Novartis. Wenping was awarded the ISOP Innovation Award in 2018 for his exceptional innovation and advancing the discipline of pharmacometrics. He was named Novartis's IVA, which is an award to celebrate his work and his contribution towards research and development. Besides his essential contribution to several NDAs across multiple disease areas, the primary reason for his IVA award is his contribution towards nonlinear mixed effects modeling software, NLMixR, which is an open source package for R. NLMixR builds on Wenping's RxODE package, a tool for simulating PK/PD models using ordinary differential equations. Welcome, Wenping, and thank you for taking your time and sharing your knowledge with us today. So let me stop sharing my screen so Wenping can start. The floor is yours, Wenping.
Very good. Thanks a lot, Aruna. Can you see my screen and can you hear me?
Yes.
Very good. Excellent. Thanks a lot for the kind introduction. And this is the outline of the workshop that we presented last week. We started with RxODE basics, and we discussed regimen simulation. And we also touched on population simulation. And today we're going to have some fun with Shiny, and we finish with some advanced topics. But before I move on to Shiny, I want to come back a little bit to recap what we discussed briefly last time. So when you use RxODE, before you do the simulation, you need to load up this library by calling library RxODE. Then you use this function RxODE to define your differential equations and to compile your differential equations. Once you compile your differential equations, you need to provide the system parameters. The system parameter should be a named vector. And another thing that's very useful is this concept of an event table. Event table is a container where you can define your dosing schedule and your sampling schedule. Once you have that, you can use this function solve with this parameter that you defined with the dosing and the sampling information that you have. And another thing we touched upon briefly is NLMixR, RxODE has built-in population simulation. In this case, let's say I'm going to simulate 100 virtual patients. Once you do this part, if you take a look of this object, this is the data frame with 100 virtual patients' PK parameters. And once you have that, you define your dosing regimen. Now, the simulation part is virtually the same as the single subject simulation. Instead of just one subject, a vector, now it is a data frame with rows representing the virtual patient's PK parameters. You run this for this 100 patients, you can see the simulation is very fast, and you can plot it out. Actually, you noticed the plotting takes more time than the actual simulation. So now it is the simulated 100 patients. All right, now let's move on to Shiny. For Shiny, there are a couple of interfaces. Both of them, they're quite interesting, but depends upon your need. You may decide to use either one of them. Let's use the first one, Rx Shiny. So here, if I type Rx Shiny, I run this one. If you have used Berkeley Madonna, then this Rx Shiny gave you a Berkeley Madonna type of simulation. So here you define your differential equations. Then you say compile. You notice once the compilation is finished, now it has the, you can change the PK parameters, you can change the initial value, and you can change the dosing, and afterwards you can say, update the plots. It shows you, in this case, a single dose. And as we did before, you can say repeat 5 times with dosing interval equal to 24 hours. Then you say update, and it gave you this 5 doses concentration profile. And there was a question before whether you can fit using RxODE to do data fitting. In theory, you cannot use RxODE to do fitting directly. But in this case, you could try different parameters and see, well, whether the concentration curve is to your liking. You can change all those things and look at what's the changes in the concentration curve. Okay, so this is the Rx Shiny part. Yet another one that's very useful is this called Gen Shiny app template. What this does is it creates a kind of a very simple Shiny application for you on the fly. You can define your differential equations here, and you can say what's the parameters you want to have, the initial values, etc. So let's run this. I say, oh, before I doing that, I need to close the other one. So let me do that again. You noticed after I run this piece of code, it says a template Shiny app has been created. We translate your PK/PD models into C and compile that. let's take a look. So I'm going to open up an Explorer. Now you noticed there's a folder called My App has been created. If you double click on that, you notice that this RxODE has created several files, including your the C code produced with your differential equations, as well as some R data objects and two files. One is called UI. If you use the Shiny, this is the place you define the user interface. You can modify that according to your need. This server part is the calculation is carried out. So you can define what are you going to do with different actions by the user, and what are the calculations you wish to carry out. And with this one, with this generated Shiny app, I can use this function called run app. Now you see I have a very simple Shiny app. You noticed now it's actually in this case I already did the modification. Let me redo this part. Then we start from the beginning. I click. I remove those. I'm going to remove this My app. All right, let's rerun this thing. Load RxODE and Shiny and call this function gen Shiny app. We come back, you noticed this My app is created. Now we run this one. You noticed by default, this Shiny app, very simple Shiny app, gave you the first compartment, the amount, and you can choose to plot which compartment. And you can say, in this case, I have QD. I can change that to EID. And you noticed when you do the changes, this Shiny app changes accordingly. And I can change that to different regimen. And then now you have 3 weeks on, 1 week off. So this template, although it's very simple, actually RxODE does a lot of hard work for you, including generating the binary ODE solver, as well as this some sample code for you to be based on your true Shiny application. So let's move on to some advanced concepts. There are certain things we touched on before. If you remember early on when we discussed RxODE, I asked you to pay attention to this sentence on line 11. So EFF0 is equal to so and so. What it means is the initial value for this EFF compartment is equal to K in over K out. So this EFF0 is an attribute, is an additional attribute that you can specify for certain compartments. And you can do the same thing with other attributes, including bioavailability. Then you use this keyword F, then whatever the compartment that you want to modify the bioavailability. If you have a lag time, you use this keyword lag to define the lag time of a compartment. And for some advanced modeling and simulation, you may wish to define the rate or model the rate or duration. Then you use the word rate and do to define these attributes. And the initial value, as we discussed before, that's yet another one. So let me give you some example how to use this. First, let's take a look at this bioavailability. And incidentally, there was a question before last time about the weight-based dosing. How should we do that with RxODE? There are at least a couple of ways of doing that. I'm going to show you both ways and also to show you the equivalent. Okay. So we're going to do weight-based dosing. One way to do that is you say, my bioavailability is equal to weight. Okay, let me get to the parameter. Compile those. I'm going to have another differential equation. In this case, I'm going to change the bioavailability of the first compartment. On this one. Okay. Now I'm going to define the parameter to be the first one. Then define an event table. There are two ways. The first way is, you noticed I have this parameter called theta. I use theta to multiply the dose. So this is one way to do the weight-based dosing. I'm going to call that the simulated object as X1. If you take a look at this X1, so it has time, the defined derived variable C2 and C3, as well as all the compartments that we have defined. In addition to that, let me use the second approach. So the first approach is multiply your weight to the dose. Yet another one, the second approach is I change the bioavailability to the body weight of the patient. For this one, you notice I have a different RxODE object. I call that M2. When I run this simulation using this M2 with bioavailability, modified bioavailability equal to the body weight. Now I change this, I run this simulation. Now I want to know the first approach is using this dose multiply by your body weight. The second approach is to change the bioavailability by the body weight. I want to see if they gave me the same simulated results. Now you can see these two approaches, they are identical. So therefore, this attributes for the bioavailability with this keyword F is yet another way to modify certain attributes of your compartment. We mentioned about this lag time and duration and rate. And let's take a look at another example using these advanced attributes of a compartment. Let's take a look at this one. So I'm going to define another set of differential equations. So it's simply a two-compartment model with absorption. But now I will have split bioavailability. F2 goes to the central compartment. The 1 minus F2 goes to the depot compartment. Further, I'm going to say there will be an infusion. The infusion is equal to a parameter. I call that D2. And in this case, what I want to do is I want to do a simultaneous first order absorption and zero order absorption. So I want to get your attention to these three sentences that we have here. So the rest actually is pretty simple. We compile this with a call of the function RxODE. As before, we define the parameters, this helper function to solve the differential equation, and then plot out the second compartment, the C2 there. Now, I'm going to have two at dosing. One is this infusion. This infusion goes to the central compartment, the second compartment. You noticed now the rate, if you use the Nangman, rate is equal to -2. What this means is we are going to define the duration of the infusion. So duration of the infusion will be a parameter. In this case, it's D2. Because we use D2 to define the duration of the infusion, so therefore, in our parameter definition, we need one more parameter for this D2, in addition to this F2 regarding this first order absorption and zero order absorption split. And of course, we also have this first order dosing. It's the same as we did before. By default, it goes to the first compartment. Now we run this code. What you have here is a simultaneous zero order and first order absorption with 5 doses. So with that would be our workshop. What I want to do next is I want to give you some exercise for you to go back and maybe practice by yourself. So the first homework actually is very simple. Aruna and her colleagues will send you these example files. I would like you to do the simulation with this ex1.r and ex2.r. And also make sure you understand what we're trying to do there. If you can do these two, I would say maybe 60, 70% of the user scenario of RxODE, you should be able to do that. The rest of that, especially the exercise number 4, will be more advanced. For the second exercise, what I want you to do is to use this Gen Shiny app template to produce a skeleton Shiny app. Afterwards, I would like you to do some modifications. First, I would like you to modify the UR, the produced UI.R and server.R, and plot out the concentration from C2. If you remember, by default, it only plot out the amount of the first compartment. In this case, it's the depot compartment. So what I want you to modify that is to plot out the concentration from the central compartment. Another sub-exercise I'd like you to try is, I want you to modify this server.r to produce some population simulation. Let's say you're going to simulate 20 subjects. And yet another one. So in this code, we just went through, we did simultaneous zero order and first order absorption. What I'd like you to do is, based on this example, you're going to do a sequential zero order and first order. What I mean by that is, you want to do a zero order absorption first, then the first order need to wait until the finish of the first order, the zero order, then you start your first order absorption. And the last exercise will be a little bit more advanced. Let me discuss this one a little bit with you further. And if you have the chance to read really the fantastic paper by Don Major and Wojciech on this quasi-equilibrium approximation in this TMDD, target-mediated drug disposition. They will give you the formula how to use the full TMDD model. You can simplify that using this quasi-equilibrium assumption to have a reduced set of differential equations. For the full TMDD, you'll have 4 differential equations with this equilibrium, quasi-equilibrium approximation. you have these three differential equations. The assumption there is you're assuming this getting together of this receptor with this ligand, as well as dissociation. This process has been reached steady state or equilibrium. So therefore, you define this KD, which is equal to this K off over K in. And because of that, you can solve this concentration, which is this funny equation. Then with all those equations, I would like you to use some parameters. I can synthesize some of the parameter values, but I want you to implement both this full TMDD differential equations as well as this quasi-equilibrium TMDD equations. I want you to see how good the quasi-equilibrium approximation is. So here on this slide, I showed you this full TMDD. So what I want to do is, based on this full TMDD and based on this differential equation here, as well as this QE assumption, as well as based on the QE assumption, what would be your drug concentration C? Based on those formulas, to also evaluate the concentrations based on this quasi-equilibrium approximation. And if these are the parameters that you're going to use, this is the difference. This is the event table you're going to use. If your implementation is correct, you would see a plot as I shown you here. So this the solid lines, they are from your full TMDD model. The dashed lines, they are from your quasi-equilibrium approximation model. As you can see, for this set of parameters, this quasi-equilibrium works really well. So that would be your bonus point if you can do this one. So Aruna, let me stop here and see if should we go on with Q&As, or if you have any comments or suggestions at this moment.
Thank you, Wenping. So I think we can take on, I did have a couple announcements here. I do see that there are some questions and requests that we are getting. I think one of them has been that some of the people who are attending the workshop may not have been getting emails with regards to the previous recording, day one's recording, the homework material, and any communication that is coming out from Project Dontabhaktuni. I would request you to please reach out to us via email to dontabhaktuni.org@gmail.com. You can also follow us on LinkedIn. That way you will get all the material has been posted on LinkedIn as well, and you should be able to see all of that. And definitely send us an email. This could be because of a firewall or how your email server is set up. And our commitment is that you get all this material and you're able to work on these homework assignments and these exercises. And Wenping, we have some questions that have come up in today's conversation.
Yeah.
I think we can get started. I mean, have you covered the material that you wanted to cover? Would you want us to jump into question and answers now?
Yeah, let's go to the question and answers. I have a copy of previous questions. Should we start with the questions before, then come back to today's questions?
Yes, that would be a great idea. Thank you.
Okay, very good. So here are the questions that you guys have raised. The questions we already covered. I will not go over that again regarding, for instance, how to add variability, and also how does RxODE knows where the infusion goes, etc., etc. So today we also covered this weight-based regimen. So I assume that's answered. Next question is, can RxODE do fitting to data and fitting using a two-compartment oral PK model to estimate PK parameters? RxODE by itself doesn't do parameter fitting. So there are a couple of ways of doing that. One is there is the sister package called NRMixR. And NRMixR is a package dedicated to model fitting, including population model fitting, as well as single subject concentration or single subject data fitting. And this question is, RxODE scientific value sensitive? Seems the theta are all in scientific value. I'm not totally sure what this question means. So I'm going to skip this. Whoever asked the question, yes.
Hi, Wenping. I think they were talking about the values, theta values given in scientific notation, not in a regular decimals. It was given in, scientific notation, like 3.0 e to the.
Oh, got it. Thank you. Thank you for that clarification. The question is, you do not have to use scientific expression or happen with. Okay. So in this case, indeed, it's a scientific expression. Well, you do not need that. You can just say 0.294. It's perfectly fine. As a matter of fact, once you run this thing, you notice R internally just convert that to your ordinary kind of floating value. Next question, how is RxODE comparable to MRG solve? Well, this is an excellent question. Indeed, in terms of functionality, MRG solve and RxODE, they are very similar and comparable. However, the design philosophy is different. MRG solve, the philosophy is if you use NumMem, you're familiar with how NumMem does simulation, you will find MRG solve to do simulation to be similar. Another thing is, I believe, I myself, I never used MRG solve before, so what I say here may be wrong. What I believe is, in order for you to define differential equations, the user may need to write some piece of C++ code versus RxODE. RxODE use this kind of, in my opinion, fairly intuitive syntax to define differential equations. And because of that, the implementation of RxODE is a little bit more complicated. We need to do parsing if we use the computer science terminology. But there are a lot of advantages for doing this parsing. Because of that, we can do code generation, which when you do model fitting, let's say the population model fitting using anomics-R, that parsing turns out to be essential. So that gives RxODE some advantage over MRGSolve. Next question, curious, why normal distribution? This question, I'm again unsure. If some of you have additional insights, you can help me out. If not, what you have here, maybe if you're talking about this R norm, indeed, this is a normal distribution. But you noticed I exponentiate this normal random variable. Because of that, now this has the distribution of the so-called log normal distribution. Log normal distribution has this characteristic of long tails. It's always positive, but it's skewed to the right. And people noticed in biology and in pharmacology, this long-tailed, skewed to the right distribution is more realistic about the population parameter distribution. How can we translate EVID 0, 1, 2, 3 from NumMem to RxODE? Again, whoever asked this question, I want to thank you. That's an excellent question. The way you do that is, we touched on that quickly towards the end of last week. There is a very useful function called s.et. ET stands for event table. So what you do is, if you have a NumMem data set with EVID equal to 1, 2, 3, 4, then you can use this s.et to translate a NumMem data set into a RxODE event table, and the rest will be the same. How is the simulation interval determined? Again, I'm not positive about what you mean by this. There is this dosing interval, of course, 0 to, now we say every 24 hours. But the simulation interval actually is not defined. What you want to do is add sampling, this kind of thing. You are going to define how long you are going to do this simulation. So let me quickly show you this part. So in this case, you see I simulate a profile from 0 to 24 hours. I can easily change this to 0 to 48 hours. If I rerun this thing, now my simulation interval is 0 to 48. So therefore, it is up to the user to define how long you are going to follow the simulation, and how often you want to sample the simulated package. Could you add a seed so that each time you can have the same result? The simple answer is yes. You can use a function in R called set seed. If you do population simulation, this is very handy to have a seed so that later you can reproduce your simulation. Excellent question. Following Vivian's question, can we have named assignment for dosing? I do not know what this question means. So maybe whoever asked this question, you can reach out to me. I will clarify that with you later. I would also like to know if Wenbin can share some example how RxODE works with Puppet. If it does, my short answer to that is I do not know how to do that. I'm not a Puppet user, but presumably, if you have you need to do simulation. If I don't know, my short answer to that, I do not know. So maybe let's move on to today's questions, Aruna.
Yes, Wenping, we can do that. And Rajan, can I request you to go through the questions and present?
Yes, Aruna. Yeah.
Please.
Okay. So, something on recordings availability and all that. Yeah. One question is can RxODE can be used to generate visual population on ODEs that are time dependent? For example, having one compartment model with something like an equation like CDT time, then where E is only activated for a small period of time within a time window. So time dependent.
Yes. Yeah. That's an excellent question. The question is, can the parameters be time dependent? The answer is yes. We didn't cover that because of time. One thing you can do is, let me go back to the slide deck. In the slide deck, I mentioned to you, there are some online resources. In particular, what I want to have your attention to is this reference number 4 here on slide number 3. There, you will see examples of time-dependent parameters. So that's the one way you can use time to define your time-dependent parameters. Yet another one is, I mentioned to you that I mentioned to you before, you can have a non-MIME data set, then you use this S.ET to convert a non-MIME data set to a RxODE event table. In your non-MIME data set, you could have time-changing covariates. And if you use the time-changing covariates anywhere in your differential equations, then this S.ET will understand you need that. And then you can use that to do your simulation as time-changing covariates or time-changing parameters. Okay.
Okay. Yeah. So I'll go to the open questions. So there is this one we have also noticed the installation keeps failing, and the RxODE not available for the version 3.5.1.
Inflation, sorry, could you repeat the question?
Sorry, installation keep failing.
Yeah.
That is
Yeah.
the RxODE not available for the R version 3.5.1.
I see. Okay. Indeed. So this is a common issue with this open source R, is the version control part. What I encourage you to do is, let's see, again, to this online website. So this one is GitHub, Anomic SAR Development, RxODE. Let me show you what you can do there. So you copy this second link, you go to your browser, so go to, this is our Anomic SAR GitHub website. So you select Anomic SAR, and another thing you can do is you go to the releases, and in the releases, okay, under this one, Anomic SAR release version 1.1.1-3, there is this installation file. I recommend you use this one to do installation, then you can avoid some of the struggle when you do installation by yourself. Next question. Can we go to next question?
So I think there is another.
Sorry.
Yeah.
Yeah.
Raj, do you want to read out the
Yeah.
question?
Yeah. RxODE, whether it can be able to stimulate NP-based drug formulation to predict concentration in a particular compartment, organ, say brain, from the central compartment. Any specific fact to be considered for that such kind of simulation?
Could you repeat the question, please?
Whether RxODE will be able to simulate NP, I'm not understanding, NP based drug formulation to predict concentration in a particular compartment, organ, say brain, from the central compartment.
Okay. Okay, got it. I think the question is maybe you have I'm guessing the question. Let's say you need to do physiology-based PK, and you want to output the concentration from different organ compartment. Whether RxODE can do that. The quick answer to that is yes, absolutely you can do that. So let me show you a couple of things, what I mean by that.
Venting?
So, yes.
Sorry, we have 1 minute to 11 now.
Okay.
So can we have all these questions answered offline, and we will share the answers that Wenping would be sending with the rest of the audience. And I see that it may be a good time for us to close the meeting here. Again, thank you, Wenping, and my apologies that we were not able to go through all the questions. I see there are several more here. And we will have them answered by Wenping and have the answers sent to you via email. Again, I just wanted to take 1 minute to acknowledge our team, which has made this workshop possible. Again, a workshop of this magnitude would not have been possible without a huge team in the background working with us. First of all, I want to acknowledge Wenping for taking his time and sharing his knowledge with this broad audience. Thank you so much, Wenping. I know I have been a pest asking you to talk about this for years now. Thank you for accepting my request. And,
thank you.
Sure. And I would like to thank ISOP and Enrico in particular for being the liaison and co-hosting this workshop with Project Dontabhaktuni. Thank you for everything. And Ramya, Krishna, they are the interns with Project Dontabhaktuni. They have been working tirelessly to get you guys access to all the communications, and Ramesh Naidu, who's part of PharmaPro Consulting, has also been working tirelessly to make sure that we can keep all of you guys posted and keep in loop. Rajan, who has been instrumental in and been my right hand to cause all of this, and I apologize that I'm 2 minutes over now, but you know, I really want to take time to thank all these people who have played an important role. And of course, our sponsors, Ramesh McKenna and Mohan Kumar from CRC Pharma. Thank you for supporting these workshops, and thank you for causing us to make these workshops available to all of you. And we look forward to seeing you in our future workshops. Again, as I had mentioned, we are going to be having another workshop coming up in August. You will hear more from us on LinkedIn and other social media as well. Look forward to seeing you in our future workshops. And as I told you, we will follow up with homework assignments that you have seen in today's workshop. And we will follow up with the mentoring program as well, so we can support you in getting the knowledge that you wanted to earn by being part of these workshops. Thank you all, and thank you, Enrico.